viral vectors have a limited packaging capacity and cannot be used to deliver gene constructs larger than 5 kilobases. To address diseases caused by mutations in large genes, dual AAV vectors are used. The coding sequence is split in half, with each half containing a complementary recombinogenic sequence to mediate recognition and recombination. Each half is packaged into a separate AAV vector. Both vectors are co-delivered by subretinal injection to co-infect the same target cells, delivering the front and back half coding sequences. Once both vectors have entered the nucleus and their genetic materials are released, the respective halves of the delivered gene recombine to form the full-length construct that ultimately generates a functional mRNA and produces a full-length therapeutic protein. Subretinal injection is a standard method for delivery of adeno-associated viral vectors encoding therapeutic genes. However, transduction with conventional AAV vectors is usually confined to the margins of the original injection bleb. This complicates the administration of vector to foveal cones, since this region of the retina is especially prone to damage upon surgical detachment. Athena's novel AAV SPR vector spreads laterally well beyond the margins of the subretinal injection bleb. This allows for safe and efficient transduction of foveal cones without the need for surgical detachment, as well as for transduction of larger areas of the retina. Both features represent distinct advantages over currently available vectors and will potentially improve efficacy and safety in patients.
Of nervous system excitability leads to severe neurological diseases, such as epilepsy. Epileptic seizures originate from specific brain areas, but current treatments affect all cells, leading to side effects. Epilepsy and other brain disorders are caused by altered neuronal firing, which is controlled by ion channel proteins that function to either increase or decrease neuronal excitability. Redpin uses a chemogenetic approach to develop ligand-gated ion channels as a drug-controlled gene therapy that can be delivered to neurons, allowing the neurons to be either activated or silenced in the presence of the small molecule drug varenicline, an anti-smoking agent that is approved by the US Food and Drug Administration. The engineered ion channel gene is focally delivered to the disease-causing neurons with cell-type specificity using an adeno-associated viral vector. The gene then selectively expresses the chimeric ion channels in these neurons. The expressed ion channels are inert in the absence of varenicline. Varenicline is taken orally and activates Redpin's ion channel receptors in the targeted neurons, stimulating or silencing their activity to treat seizures. This approach can also be extended to many other neurological disorders. Redpin's chemogenetic approach merges gene therapy with pharmacotherapy and is poised to have a transformative effect for treating currently intractable neurological diseases.